Hello, my name is Marcelina Bednarchuk and I am a PhD student working with mRNA decapping enzymes. Today, I would like to introduce you to my research project concerning inhibitory studies on decapping enzyme from Vaccinia virus. As you probably know, the degradation of the messenger RNA plays an important role in many biological processes, including gene expression. Properly functioning eukaryotic decapping enzymes like DCP2 regulate mRNA decay in a right way. But it turns out that some viruses also possess the decapping machinery that helps them to avoid the host's innate immune response, as in the case of poxvirus representative, Vaccinia virus and its D9 decapping enzyme. The main aim of my PhD project is to develop new, effective method to study activity of D9 enzyme and thus better understand the role of decapping enzyme in viral infection. To do this, we use the fact that D9 recognizes 7-methylguanosine and we checked if the probe containing M7G moiety and the fluorescent tag would be cleaved by D9 and if we could observe an increasing fluorescence signal during hydrolysis process. After obtaining positive results, we tested 5 M7G probes with different length of phosphate bridge and different fluorescent dyes attached, and selected one, M7GTP labeled with pyrene, with both highest fluorescence response and good hydrolysis kinetics. In the next step, we apply this approach into HTS mode to provide fast and effective screening studies to identify small molecule compounds as potential D9 inhibitors. We validated our developed method by determining the kinetic parameters of the hydrolysis process and determining Z-factor parameter, which value 0.75 confirmed that the method could be used in high-throughput screenings. Then, we use the optimized and validated assay to evaluate our in-house library of nucleotide-derived compounds, identifying some inhibitors with nice nanomolar IC50 values. This method is also suitable to screen commercially libraries like LOPAC. Once again, those studies enabled us to identify several pharmacologically active compounds with inhibitory properties against D9 decapping enzyme. Finally, we verified the inhibitory potency of the most promising hits from screening experiments on short-capped RNA substrate in vitro decapping assay. The obtained data were consistent with the results from screening experiments, and this ultimately confirmed that our method works and gives reliable and reproducible outcomes. To shortly sum up, we present you a novel, simple fluorescent method to monitor D9 enzyme activity that can be used to very efficient and high-throughput inhibitory screening experiments. We demonstrated the potency of the method by screening our in-house library of nucleotide-derived compounds as well as commercially available LOPAC library and confirmed identified hits with different independent methods. And the most important thing, we believe that this method can be successfully applied to study another decapping enzymes as well. Thank you for listening, and I would be happy to answer any questions.